Glycolysis. So, glycolysis is a metabolic process that serves as a foundation of both aerobic and anaerobic respiration. In glycolysis, glucose is converted into pyruvate, and glucose is a six-membrane ring molecule found in whole blood and is usually a result of a breakdown of carbohydrates into sugars. It enters the cell through a specific transporter protein that moves it from one outside to the cell into the cell cystole. All of the glycolytic enzymes are found in the cystole. Now, the step 1, hexokinase. So in this first step, here the glucose ring is phosphorylated, and phosphorylation is the process of adding a phosphate group into a molecule. So in a molecule derived from ATP. As a result, at this point in glycolysis, one molecule of ATP has been consumed. So the reaction occurs with the help of the enzyme known as hexokinase, and an enzyme that catalyzes the phosphorylation of many six-membrane glucose-like structure. Atomic magnesium is also involved to help shield the negative charges from the phosphate groups on the ATP molecule. The result of the phosphorylation is a molecule called glucose 6-phosphate, thusly called because of 6 carbon of the glucose acquires the phosphate group. Now the step 2, phosphoglucose isomerase. So in this second step of glycolysis involves the conversion of a glucose 6-phosphate to fructose 6-phosphate. So in this reaction occurs with the help of the enzyme phosphoglucose isomerase. As the name of the enzyme suggests, this reaction involves isomerization reaction. So in this reaction involves the rearrangement of the carbon bond from the transformation of the membrane into a 5-membrane ring. The rearrangement takes place when the 6-membrane ring opens and then closes in such a way that the first carbon now becomes an external ring. Now step 3, phosphofructokinase. So in this phosphofructokinase, magnesium as a cofactor changes fructose 6-phosphate into fructose 1,6-biphosphate. So in the third step of glycolysis, the fructose is converted to fructose 1,6-biphosphate, similar to the reaction that occurs in the first step of glycolysis. A second molecule of ATP provides a phosphate group that is added on the fructose 6-phosphate molecule. The enzyme that catalyzes the reaction is phosphofructokinase PFK. As in step 1, a magnesium atom is involved to help shield negative charges. Now the step 4, aldolase. In this step, utilizes the enzyme aldolase, which catalyzes the cleavage of fructose biphosphate to yield 3 carbon molecules. One of these molecules is called glyceraldehyde 3-phosphate and the other is called dihydroxyoxygen phosphate. Now step 5, triosylphosphate isomerase. The triosylphosphate rapidly interconverts the molecules dehydrogenase phosphate and glyceraldehyde 3-phosphate. GAP is only molecule that contains in glycolytic pathway. As a result, all of the DHAP molecules proceed are further acted on by the enzyme triosylphosphate isomerase, which recognize the DHAP into GAP so it can continue in glycolysis. At this point in glycolytic pathway, we have 2 3-carbon molecules, but we have not yet fully converted glucose into pyruvate. So in step 6, glyceraldehyde 3-phosphate dehydrogenase, 2 main events take place. Glyceraldehyde 3-phosphate is oxidized by coenzyme nicotinamide adenine dinucleotide. The molecule is phosphorylized by the addition of a free phosphate group. The enzyme that catalyzes this reaction is glyceraldehyde 3-phosphate dehydrogenase. The enzyme GAPDH contains appropriate structures and hold the molecules in a conformation such that it allows the NAD molecule to pull a hydrogen off a GAP, converting the NAD to NADH. The phosphate group then attacks the GAP molecule and releases it from the enzyme to yield 1,3-biphosphate 1,3-biphosphoglycerate NADH and hydrogen atom. Now, step 7, phosphoglycerate kinase. Phosphoglycerate kinase transfers a phosphate group from 1,3-biphosphoglycerate to ADP to form ATP and 3-phosphoglycerate. In this step, 1,3-biphosphoglycerate is converted to 3-phosphoglycerate by the enzyme phosphoglycerate kinase. Again, this is a kinase. This reaction involves the loss of a phosphate group from a starting material. The phosphate is transferred to molecule of ADP that yields our first molecule of ATP. Since we actually have two molecules of 1,3-biphosphoglycerate, we have 3 carbon products from stage 1 of glycolysis. We actually synthesize 2 molecules of ATP at this step. With the synthesis of ATP, we have cancelled the first 2 molecules of ATP that we used, leaving us with a net of 0 ATP molecules up to this stage of glycolysis. Again, we see that the atom of magnesium is involved to shield the negative charges of the phosphate group of the ATP molecules. So, step 6, phosphoglycerate mutase. The enzyme phosphoglyceromutase relocates the P from 3-phosphoglycerate from the third carbon to the second carbon to form 2-phosphoglycerate. In this reaction mechanism proceeds by the first adding an additional phosphate group to the second position of the 3-phosphoglycerate. The enzyme then removes the phosphate from the 3rd position, leaving just the second phosphate, and thus yielding 2-phosphoglycerate. In this way, the enzyme is also restored to its original phosphorylated state. Step 9, enolase. The enzyme enolase removes a molecule of water from 2-phosphoglycerate to form phosphonol puric acid (PEP). In this step involves the conversion of 2-phosphoglycerate to phosphonolate PEP. The reaction is catalyzed by the enzyme enolase. Enolase works by removing a water group or dehydrating the 2-phosphoglycerate. The specifically enzyme pockets allow for the reaction to occur through a series of steps too complicate to cover here. Step 10, pyruvate kinase. The final step of glycolysis converts phosphonol pyruvate into pyruvates with the help of the enzyme pyruvate kinase. So again, it is a kinase reaction. As the enzyme's name suggests, this reaction involves in the transfer of a phosphate group. Phosphate groups attached to the second of the carbon of a PEP is transferred to a molecule of ADP yielding ATP again. Since there are 2 molecules of PEP, here we actually generate 2 ATP molecules. So after